So,
lovely. So shall we kick it off? So thank you so much, everybody, for making time for coming to see what Will has to talk to us about on the fourth marker for sickle cell that he's just found. He's just hot off the press. He put it up on bioRxiv the day before tomorrow. Is that the day before yesterday? Or was it yesterday? No, the day before yesterday. And he will also be talking about that in ASTMH on Sunday morning at ASTMH. So if you are at ASTMH and if you want to talk to him in person, please, yeah, great to see you too, Ellen. And please make sure to come and see Will and to join his talk on Sunday morning. So I will let Will introduce himself, but he will be a very well-known face to many of us working on the malaria field. He's been working with Dominic for many years since his PhD. He has a joint appointment as a senior staff scientist here at Sanger, and also as a clinical research fellow at Addenbrooke's, and I'm sure I messed up with your titles, Will, so I'll let you do the proper adequate work for that. I'm really sorry. But Will is one of a kind, and he holds really excellent science, and he's also a clinical medical doctor in Addenbrooke's, but nevertheless he always manages to amaze us with the wonderful science that he does. And I'm sure today he's just going to do us amazed in any way. So Will, off to you.
Great. Thank you so much, Christina, for that very generous introduction. That was so generous that it sets the bar very high that I now have to live up to. But can everyone hear me okay, first of all? Someone just give a thumbs up. Yeah. Okay, so I'm just going to share my screen. Can everyone see the proper slides? Great. Okay, so today I'm going to be talking about interactions between the Plasmodium falciparum genome and sickle hemoglobin. I don't need to give much more introduction. I think I have these different titles, but basically I'm a clinical academic, so I combine my time between clinical infectious disease medicine and pathogen genomic epidemiology research, and I worked with Dominic Kwiatkowski for a long time. So given I'm a clinician, I thought I would start with a clinical case. This is a case of a 28-year-old West African man who presents to a clinic in Ghana complaining of 4 months of a painful, non-healing ulcer on his left ankle, which you can see the slightly grainy picture of. And he also complains of bone pains, and you notice he's short for an adult, pale and jaundiced. And this would be quite a typical picture for someone with an inherited hemolytic anemia, such as sickle cell anemia. And so what is sickle cell anemia? So sickle cell hemoglobin is caused by a single point mutation in the 6th codon of the beta globin gene. And beta globin is one of the key components of adult hemoglobin, which carries oxygen inside red blood cells. And this single change has a quite profound effect on the structure of hemoglobin and of the red blood cell. So you can be heterozygous, which is when you have one of the wild type or A copies of the hemoglobin of the beta globin gene and one sickle copy, or you could be homozygous, which is when both copies from mum and dad are both the sickle copy. And in the homozygous state, you suffer from sickle cell disease. Whereas in the heterozygous state, we now know you have strong malaria protection and little or no pathology. So what is the pathophysiology of sickle cell disease? Basically, in low oxygen conditions, the sickle hemoglobin polymerizes, and that makes the red cells kind of crumple up, and rather than forming the nice biconcave disc of the red blood cell, they form this characteristic misshapen and stiff sickle structure, which you can see in the picture. That gets stuck and damages the endothelium, causing inflammation and occlusion within the microcirculation. And that causes these vaso-occlusive crises, which can occur all over the body. So it's very much a multi-system disorder. It can predispose to strokes, acute chest syndrome. It can affect the liver, the spleen, the bones, and as in the case of the clinical case that I opened with, it can affect skin and soft tissue as well. And you can get chronic ulcers. So a huge a range of problems, very challenging for healthcare, and in the absence of high quality, long-term, high intensity healthcare, unfortunately, people with sickle cell disease can often not do very well, and it can be a significantly life-limiting disease. Which raises the question why you would have a fairly high prevalence, relatively high prevalence of something that in the homozygous state can be so deadly. This is the distribution of hemoglobin S and C in Africa, and it, particularly for hemoglobin S, on a broad global scale, overlaps somewhat with the distribution globally of malaria. So 80% of individuals born with sickle cell anemia live in sub-Saharan Africa, where 90% of the deaths caused by Plasmodium falciparum occur. Hemoglobin C has a slightly more limited distribution in Central West Africa. So you can see particularly in sort of northern Ghana, Burkina Faso, Mali, compared to a slightly broader geographic distribution for hemoglobin S. And on that question of why you have this, these polymorphisms fairly widespread, this was thought about quite a long time ago. The so-called malaria hypothesis was, there's a bit of contention exactly who and when proposed it, but widely accredited as JBS Haldane in 1948. And noting the deleterious effect of the homozygous sickle cell anemia, hypothesized that maybe there was an advantage to being heterozygous, and given the overlap geographically with malaria, and the fact that malaria parasites infect the red blood cell, maybe it protected against malaria. And the direct evidence for that came quite shortly after by A.C. Allison in 1954 and 55, who showed that there was a lower proportion of sickle heterozygous children that had parasites compared to the wild type hemoglobin A carriers. So if you look, that's the primary data from that paper, that among the sicklers, which were actually heterozygotes, he found 28% had parasitemia compared to 46% among the non-sicklers, the wild type hemoglobin genes. Now, of course, we have very, very robust evidence for the protective effect of sickle. So this is data from one of the flagship Malaria genome-wide association studies on the human genetic determinants of severe malaria. 17,000 severe malaria cases versus 22,000 controls, which is, kind of unbelievable from Allison's first paper in 1955 to what could be accomplished, 60 years later. And you can see various hits along the genome, and this big signal here is from the beta-globin locus. This is from another of the malaria gen papers, the reappraisal of malaria resistance markers, separating by cerebral and severe malarial anemia in red and blue dots, respectively. This is for the beta-globin locus. And you can see that for both cerebral malaria and severe malarial anemia, in all of these places, sickle just has an incredibly strong association with protection against severe malaria. And I'll always remember reading the p-value from this paper. So this was an odds ratio of 0.14, which is a very impressive effect size, and with a p-value of 1.6 times 10 to the minus 225, which I think it might be not far off 1 divided by 10 raised to the power of the number of atoms in the universe, something like that, in terms of the size of that p-value. So very, very convincing and strong evidence of the effect of sickle. That obviously begs the question of what the mechanism of sickle protection is. And the kind of long and the short of it is basically, even after all these years, we don't really know. And I've just listed a bunch of kind of theories here. So it's known that the parasites don't grow well, especially in lower oxygen tensions in vitro in sickle heterozygous individuals. Maybe the sickling of infected red blood cells has an effect. There's a lot of interest in reduced expression of PFEMP1, which helps to stick parasites into the intravascular system and causes, is part of the pathogenesis of severe malaria, and that you seem to get less expression of PFEMP1 in parasites that infect sickle heterozygotes. Increased splenic clearance, increased hemolysis. So, lots of things have been proposed, and it might be that there's multiple mechanisms all acting kind of on top of each other for what mediates this protective effect. Another idea that I wanted to just touch on was this idea of the Red Queen, which I first read about when I was an undergraduate, and I always thought was a cool idea. And this is a metaphor taken from Lewis Carroll's Through the Looking Glass. The Queen says to Alice that it takes all the running you can do to keep in the same place. And that's used as a metaphor for evolutionary, well, specifically for co-evolutionary interactions. So, you know, an evolutionary change in a host may drive adaptation in the parasite and vice versa. If the parasite gains some virulence mechanism, then that exerts a selection pressure on the host to overcome it. And you get this kind of continuous co-evolution of host and parasite trying to kind of outdo each other. So you can ask, well, if humans have evolved this very strongly acting, significant genetic effect that reduces the severity of malaria, then what is the how has the parasite kind of responded evolutionarily to that selective pressure? And to rephrase that question, you could say, are there parasite genetics that interact with human genetic variation and have an effect on the phenotype? This is sometimes being referred to as the shared phenotype, which was a phrase I first read also as an undergraduate from a great book that was translated from, I think, French originally, called The Art of Being a Parasite by Claude Coombs. And so it's a phenotype in the case of a disease is not just from the pathogen, but it's also from the host, and that host pathogen interaction produces these shared disease phenotypes. So this was looked into by a great study led by Gavin Band, and by in Dominic Kwiatkowski's group, taking samples from the CP1 malaria gen study, specifically from Kenya and the Gambia. These were 5,000-ish samples from children with severe malaria. And there was already the human data, which had been looked at for the genetic determinants of severe malaria. And they looked at sequencing the parasite and looked for associations between the parasite genome and human genetic variation. And they looked at several human variants, but one of the results that really jumped out from that paper, quite remarkable finding, was these very strong associations with sickle, with HBS. And they named these loci Plasmodium falciparum sickle-associated loci, or PFSA 1, 2, and 3. And PFSA 1 and 2 were both located on chromosome 2, and then PFSA 3 was located on chromosome 11. This is another quite remarkable figure from that paper, showing that if you look at PFSA 1, 2, and 3 on the left, with whether it's present or absent, and you look at the protective effect of sickle, then it gets less and less the more of these mutations you have as you go from the top, which is parasites that don't have any of them, and you can see sickle having its strong protective effect. And as you move down towards having more of these loci, eventually, if a parasite has all of them, then the protective effect of sickle is essentially lost. So a kind of incredible result that even though we know how strong that protection offered by sickle is, it seems to depend quite crucially on the genotype of the parasite infecting that person. So I'm going to move on now to talk about a study that I've been working on. Initially, this study actually has a very long history, and Lucas Amenga has really been leading it on the Ghanaian side. And Lucas and I first started talking about this idea when we were thinking about SWGA. This was work that Christina Ariani and Sam Oyola were leading, of being able to sequence the parasite from dry blood spots. And we thought that would present a cool opportunity to look at both the parasite and the human genotype from the same samples. And so we started writing the ethics to do that in April of 2014, which I remember very distinctly because I was revising for my pediatrics final exams in the day and then writing the ethics proposal at night to do the study, which we got approval for. And we went and started sample collection in June of 2015. And sample collection continued after I left. And so from 2015 to 18, we collected these 2,000 samples where we had ethics to look at both host and parasite. And as anybody who's worked on study ethics knows, getting approval for the host part is usually more problematic than getting approvals for the parasite, which I think is totally fair enough. And it was a very educational experience for me, as indeed the whole way through this project has been, thinking about how to get informed consent from populations with lots of different languages present, how to translate things into different languages that might, are not easy to explain about host, human genetics and the kind of cultural as well as linguistic context of all that. So that was all very interesting. And the aims of this was to do a genome-wide association of the parasite, looking at ultimately, we ended up focusing on the human beta-globin locus. And by the time we were really focusing on this, the Band et al. Paper was already known. It wasn't quite out yet, but obviously we were speaking to Dominic. And we wanted to see, would we replicate the finding? This was for mild malaria. Would we find any additional associations? And was there any signals relating to hemoglobin C, which, if you remember from my map, is present in reasonable frequencies in northern Ghana, but is basically absent in the Gambia and Kenya. So those were the kind of main aims of this study. So from 2015 to 18, we have 2,100-ish blood samples that were collected from mild malaria patients. These are from clinics in northern Ghana, where Lucas has worked for a long time, and indeed where Lucas is from. Somewhat conveniently, they were all sequenced as part of PF7 and the ongoing Malaria Gen P. falciparum consortium community project. So the parasite was kind of sequenced that way. And then for the samples within the study window that we had ethics for, I then did a PCR-based genotyping of the Sickle locus, which again was convenient for me because Kirk had already designed the primers. So I just had to get Kirk's PCR working in the Sanger Institute. And because PCRs are fickle things, that did require a bit of tweaking, but we got there in the end. And so we ended up with 1,368 samples where we had both a beta-globin genotype from the PCR and capillary sequencing, and a whole genome sequence for Plasmodium falciparum that had passed the Malaria Gen quality control filters. So this is an example of the capillary sequencing traces that we get, good old-fashioned capillary sequencing, which hilariously, despite being Sanger's sequencing method, is no longer done at the Sanger Institute. We have to send the samples to Germany to be done by Eurofins. And you can see there a clear wild type position on the left, a homozygous for Sickle on the right, and then a heterozygous in the middle, which has both the T and the A allele present on top of each other. And I looked at what would have been 2,100 of these tracings in order to genotype them all as either Sickle or Het or wild type, or hemoglobin C, which is also present there. So that was a fun. You can kind of get into a good meditative state of just clicking through these samples. And so these were the end counts. And we actually had a slightly lower proportion of Sickle genotypes than I was maybe expecting. So it was a 2.2% that were hemoglobin AS and only a single individual that was Sickle homozygous. Speaking with Lucas about that, there's a number of possibilities. Two that I think are quite interesting is that you're selecting a population that has malaria. So you would expect there to be a slightly lower prevalence of Sickle than the population at large, because Sickle protects against malaria. So you're kind of filtering out some of those Sickle people, if that makes sense. And then the other factor is, unfortunately, in terms of the Sickle homozygotes, it may be that in a relatively deprived area, as this is in northern Ghana, that unfortunately the healthcare infrastructure is simply not there. And so the survival rate is poor. So this is just showing the same data, but kind of graphical, just to highlight the majority of people were wild type AA, but there was a decent proportion of hemoglobin C heterozygotes as well, and then a fairly small proportion of hemoglobin S carriers, most of which were Hets. So the next thing we did for the analysis was to assess for population structure in order to do the genome-wide association study, and also a bunch of SNP filtering from the huge number of variants that PF7 gives you from the PF7 VCF, which is like, a lot of variants, over 1 million variants. So we filtered it right the way down to 14,000 biallelic pass SNPs with at least 1% minor allele frequency and no more than 25% missingness. We did principal components analysis. We saw some genomes that were very, very highly related to each other, which are marked in red here, that we removed, leaving 1,309 for analysis, and then we recomputed the PCAs. And then for the actual GWAS, we use principal components 1 to 3 as covariants in the GWAS. You can see that components 4 and 5 were driven by single SNPs, which when I looked at them weren't very exciting SNPs, so I kind of just ignored that. And this is basically the result. So this is the kind of key slide of the whole thing, which is the Manhattan plot showing the SNPs along the 14 falciparum chromosomes and the p-values for the association. The top plot is for hemoglobin S, and the bottom plot is for hemoglobin C. Key results are PFSA 1 and 3 were very convincingly replicated in this paper. So that's on chromosome 2 and 11, that's sickle associated. And a possible new signal of association on chromosome 4, which you can see kind of marked there, putative new locus. And then somewhat disappointingly, no strong signals of association jumped out with hemoglobin C, which I have some thoughts about, but maybe I'll come back to that a bit later. But the rest of the talk, I'm going to focus on this, this new locus on chromosome 4. So this is focusing down on that in higher resolution on that bit of chromosome 4, and the associated signal is within this gene called FIC 4.2. Antoine tells me that I shouldn't say FIC because apparently that means something quite rude in German, but it's in my head now, so that's what I'm going to say. And it turns out that FICs are actually quite an interesting gene family, which I'll expand on more in a bit. But there's these two hits. One of them was more significant than the other, and they were both non-synonymous mutations in the coding sequence of the gene, which actually was quite handy for replication. I also used a completely different approach to do the genome-wide association study. This was with a linear mixed model using a fast LMM, which has been applied to Plasmodium falciparum before. As a linear model, it's intended more for kind of continuous data rather than a discontinuous sickle or not sickle. So it didn't quite apply so well, and that is why I think you get inflated p-values. But nonetheless, I was pleased that it found exactly the same signals, just in terms of reassurance of repeating by an independent method. We've got PFSA 1, 3, and then this potential new signal. So I'm going to mention another paper which we were able to use for replication, and it's also just a really cool paper. So if you fancy some bedtime reading, then you can have a look at this, which looked at the transcriptional profile of sickle cell trait hemoglobin for Plasmodium falciparum. So there's this nice plot, ever since EB, I've always liked these transcription plots of the falciparum life cycle. So as the time goes on, you see different genes that switch on and off. I think one of the headlines of this paper was actually quite similar genes were turned on and off in a similar order for both AA and AS individuals. But the paper goes on to look at differentially expressed genes between sickle and non-sickle individuals. But the reason why it was useful for us is because we could then use the RNA-seq data to look at the coding, change in that FIC 4.2 gene, which meant that we could use it for as a kind of validation or replication data set, which is what this figure shows. So this is looking at the Gavin Band's paper from, so that's the Gambian and Kenyan samples of severe malaria. And one thing to say is that this locus PFSA4 is just not present in Kenya at all. So that's why there's no estimate. And it wasn't significant in the Gambia. In Ghana, it was significant, and in Mali, it actually replicated. And you can see the meta-analysis there. So potentially, we thought, well, this is an interesting signal, and it's replicated in an independent sample set. It just didn't replicate in the Gambia for some reason, and it's not found in Kenya. And I remember when I first showed this to Dominic, and he was very excited by it, and immediately he was like, it's got to be hemoglobin C. Mali, Ghana, that's right on top of where hemoglobin C is found at high prevalence, whereas it's not really found in the Gambia or in East Africa at all. But that's just kind of a hypothesis. I don't know. I don't have any further data that can support or refute that at the moment. So the other interesting thing was that we found a very similar magnitude of effect size for PFSA1 and PFSA3. This is an example for PFSA1. So Kenya and Gambia were for severe malaria cases from CP1, and Ghana were the mild malaria cases from this study, and they're all basically the same, which Dominic was very excited about. And Dominic's view was that that must mean that these variants have a kind of general effect in allowing the parasite to get into sickle heterozygote red blood cells. And so it doesn't predispose to severe malaria per se in those people. It's just that the parasite can get in to sickle heterozygous cells and cause both mild and severe malaria. So that it's kind of subtle, but potentially important in understanding the mechanism of what's going on. This is a plot that I just quite like because I still just can't believe how giant the effect sizes are. And just as a slight digression, I mean, if you're used to reading human GWAS of, like, you know, diabetes risk or something, the idea of having 1,000 people, of which 36 had the phenotype, which is the case in this study, we had 36 people with sickle alleles present, and expecting to find anything for, you know, diabetes, cardiovascular risk, etc. I mean, it's, you know, that's just not how human GWAS are done. But that's because in human GWAS, the idea of seeing an effect size of this magnitude from a small number of genes, you never ever see that. You know, despite the Daily Mail reporting a gene for obesity or whatever, the effect sizes are usually much, much smaller. To have such large effect sizes is really remarkable, as you can see here with the frequencies of the PFSA variants in sickle and non-sickle individuals for the population in Ghana. This is some plots that I played around with for a while, and I was quite sad I never got to show them to Dominic, actually. But I don't know how much other people will like them, but in my head, I quite enjoyed them. So, what I was imagining when I made the plots was imagining things either from the perspective of a human host, or from the perspective of the parasite. So from the parasite's perspective, these are the proportion of parasites that either have or don't have PFSA in the top plot, infecting people who either do or don't have sickle. And so what this is saying is that if sickle is present, then around half of the parasites infecting that person will have sickle-associated variants, either 1, 2, or 3 around. Whereas in people who don't have sickle, the vast majority of the parasites that infect that person will not have any of the PFSA variants. And then looking at it from the other perspective, this is the proportion of sickle hosts that are infected either by PFSA or PFSA negative parasites. That's the bottom plot. And so if you're a parasite that carries one of the PFSA loci, then something like 20–25% of the people you encounter will be sickles, compared to if you're a parasite that doesn't have a PFSA locus, then a very small number of the people you encounter in northern Ghana will be sickle hosts. So I don't know, maybe that does it for you, but I quite like those plots. Maybe I didn't do a good job of explaining them. Okay, I've got some slides on FIC 4.2 now. This gene is a serine threonine kinase, and I was actually quite pleased when I looked into this gene, because so many times I've done stuff with Plasmodium, and then I get some signal and I'm excited about it, and then I look up the gene and it's unknown function. We don't know anything about this. Whereas this actually is a gene that we knew quite a lot about. So people have knocked this gene out, they've knocked it down, they've done experiments on it. This family has massively expanded in the Laverania clade. It's exported into the host red blood cell, and each of the different FIC kinases have a different bunch of host proteins that they phosphorylate, which I've heard called a phosphorylation fingerprint. And if you knock out FIC 4.2 specifically, then the parasites do have a phenotype. The red cells that are infected are less rigid, less adhesive, and in some kind of mechanism that's independent of PFEMP1. Which is all kind of cool, because that seems like a good candidate for something that might have some effect on modulating the intraerythrocytic environment of a sickle versus a non-sickle red blood cell. And then I tried using AlphaFold, which was kind of just for fun, really. I've never used AlphaFold before, but for those who don't know, this is a kind of AI program that folds up proteins based on the sequence or the predicted sequence. And like a lot of Plasmodium proteins, there are big loops where the algorithm doesn't know what to do with them because Plasmodium has these kind of weird low complexity loops. But there's some key bits, which is how we know it's a serine threonine kinase, which are that have more kind of structure. And this was the top signal variant, which is a leucine to isoleucine mutation, just next to the kinase domain of the protein. And then the second kind of highest hit that was lower association, but in quite high linkage with that other SNP, is also right in the around the kinase domain. So maybe it has some effect on the kinase activity of the gene. Again, that's just total speculation. I don't have any kind of wet lab data on that. The last part of the talk was some analyses that I did using PF7, looking at the frequencies of PFSA 1 to 4 loci. And what I did was I got all of the countries from the PF7 database that had greater than 100 QC pass samples. And I've organized them here, roughly speaking, West, Central to East Africa, and then Colombia is kind of on the end. And Colombia was a bit weird, actually. I don't know really what's going on there. And I didn't include Asia because essentially these variants are completely absent in Asia. And I think just by eye, there kind of was, broadly speaking, a pattern, which is that there's a kind of West Africa style of pattern where you have PFSA 1, 3, and 4. And then there's an East Africa pattern where you have PFSA 1, 2, and 3, but not 4, which is why it was absent in Kenya. And then there's Colombia, which I was also just kind of totally surprised by, that has actually a really high frequency of PFSA 3 and 4, which may be like some kind of weird artifact because all of the Colombian samples in PF7 come from, well, nearly all of them come from a single study. I don't know what the epidemiology is of that study. Maybe that population had a high amount of African heritage. So yeah, I was a bit puzzled by that. But what I then did was looked at linkage between these variants, and I thought for a long time about how best to present these data, and I came up with these plots, which I don't know, maybe other people won't like, but again, I have ended up staring at them for long periods of time, and I quite like them. So you've got the frequency on the left, and then the linkage as a grid on the right. And this is for Ghana from PF7. So kind of reiterating the West African pattern that you've got PFSA 1, 3, and 4, and not much PFSA 2 at all. And some of these linkage values are just absolutely nuts, which had already been reported by Band et al. For PFSA 1, 2, and 3. But like the linkage between 1 and 3, which are located on chromosomes 2 and 11, is 0.67, which if you're used to looking at linkage, that's just nuts. So actually, as a separate analysis, I just looked for what are the most linked variants that are not on the same chromosome. And these are right up there as some of the highest linkages in the entire genome. So just to expand on that for people who are not so used to thinking about linkage, sex makes genetics much more interesting, and the genome's getting cut up all the time into little pieces. And so you would expect there not to be much linkage in variants located on different chromosomes because the genome's constantly getting shuffled every generation. And yet somehow these variants on different chromosomes very often, more often than not, are found together on the same parasite, which if you assume that sexual recombination isn't doing anything weird or special with these variants, to me must mean that there's some really good reason biologically that the parasite likes to have these variants together, and that selection is removing parasites that have broken up the variants. Otherwise, I don't know how else you could explain the linkage. But anyway, PFSA 4, also pretty high linkage, although not quite as high as 1 and 3. So this, I would say, is the central West African pattern seen in Ghana and Mali. I don't think I'm showing it, but Mali has a very similar pattern. This is the East African pattern where you don't have PFSA 4, but you have 1, 2, and 3. And again, pretty high linkage values there. this is me just comparing Ghana and Kenya to kind of highlight that point. This is Colombia, which I just added for fun because I don't really understand it, where crazy high PFSA 3 and 4, 50-60% of the parasites in that study carried those two variants, and the linkage was like 0.49. So again, one of the highest linkages anywhere in the genome, and these are on different chromosomes. So PFSA 3 is chromosome 11 and PFSA 4 is chromosome 4. So that's kind of interesting and cool. If anyone wants to go and do a study in Colombia, that would be interesting. So I've got some conclusions now. So in a population with mild malaria in northern Ghana, we replicated the association between hemoglobin S and PFSA 1 and 3 that was discovered in Gavin Band's et al. Paper. And we found a new locus of association in this gene FIC4.2, which we've called PFSA 4. And that replicated in a population with mild malaria from Mali. The frequencies and patterns of correlation between the variants vary a lot between different countries, with kind of broad patterns in West and East Africa. And we didn't find any associations with hemoglobin C in this cohort. My kind of thoughts around that are that hemoglobin C has a kind of gentler phenotype on the red cell, and therefore maybe has a gentler knock-on effect on the parasite. And so maybe the effect size is smaller. And so to discover associated variants, you need a larger sample size. And so maybe you would discover hemoglobin C associations if you had a proper human GWAS style powered GWAS. So maybe it's just an effect size problem, a sample size problem. If you want to learn more about that paper, then as Christina said, it's now up on bioRxiv as of a few days ago. Just some final acknowledgements. It ended up being a very collaborative study. So, Gavin kind of joined the team, and it was really fun working with him from the Wellcome Sanger for Human Genetics in Oxford, where Kirk is also based, as well as all of the people from Ghana, from the West African Centre for Cell Biology of Infectious Pathogens, and from the Norongo Health Research Centre as well, which actually I think I need to add to this acknowledgement slide. And from everyone at Sanger. And obviously, I just want to kind of dedicate the study to Dominic Kwiatkowski, kind of in memoriam. The study is now hopefully going to be published. And with that, I can take any questions.
Thank you very much, Will. I will suggest people, yes. I will suggest people putting their hands up or writing on the chat, and then we can take the questions from there, if that's okay. So we already have a couple of comments on the chat. The first hand up is from Dominic. So what I could do is I will alternate between people putting their hands up and a question from the chat. Okay, so the first hand up was from Dominic.
Hello?
Hello.
Yeah, can you hear me?
Yes, I can.
Yes. Yeah, that's a very wonderful presentation, very impressive and deep analysis. So I followed the talk throughout, and the question that is on my mind is if these are the current pool of parasites that we are having, where they are adapting to the sickle cell, in order to be able to predict how the parasite will be like in the future, is it possible to go back a few years? Because I think this study is from 2015 to 2018 samples, I believe. Is it possible to go a few years back, maybe in the 90s or something, if there's some form of data available, and analyze it to see the kind of trajectory or evolutionary process the parasite is going through? That way we can be able to predict some of the signatures that may exist in the future. Thank you.
Yeah, thank you for the question. So I'm not sure if were you thinking specifically about the sickle associated loci, like whether they change in frequency over time?
Yes, yes, the ones in the parasite, because I know these parasites also keep changing and certain mutations keep accumulating. So your data is suggesting that they are now adapting to the sickle cell in the host, the
Yes.
sickle variant in the host. So I'm saying if we went a few years back, is it possible to be able to get a trajectory or see a pattern through which we can predict into the future how these phenotypes in the parasite will be like?
Yeah, I mean, that may be possible, especially with malaria gen data. I think the two countries that we have really good longitudinal data from are Kenya and Ghana, and maybe also the Gambia. So someone could look at how the frequencies have changed over time. I haven't done that, so I don't know for sure what you would find. But what I would say is I would be a bit surprised if there was a major trend specifically relating to sickle, because as far as I don't think I would expect the frequencies of sickle in those countries to have greatly changed. And you would think that that's what would drive a change in the frequency in the parasite population, would be a change in the frequency of the. So over the time frame of like from the 90s, I wouldn't have thought, although science might prove me wrong, that they would change that much. But what would be really interesting, although I think incredibly hard and possibly impossible, would be to go back 50,000 years, or when sickle first emerged, and then see how these changed. I mean, you can do things like you can try and do molecular clock methods to sort of date the origin of the parasite mutations and compare that with the. But even doing that with sickle, actually, just to find a date of it when people think sickle originated, turns out to actually be really complicated, because there's like loads of different studies that will come up with different dates. But yeah, I don't know if that answered your question. No.
Will Kirk would like to add something.
Yeah, just to say, when we did the study with Gavin, we did look at that because both the Gambia and the Kenyan samples go back to the late 1990s. And certainly in the Gambia, we had about 200 samples each year for 10 years. And we really didn't see a change in the frequency of any of the PFSAs. The other thing to note is like Will said, he didn't include the Asian PF7 in the data. But when you look at that, it's very interesting, the fact that these PFSAs are only present where sickle is present. Maybe that's why there's no sickle in Asia, so there's no reason why these PFAs need to arise. So yeah, it's a very interesting question. When did it arise? Why has it arisen? And, have they changed in the parasite over time? But all we can say is in a group over a period of about 10 years, maybe 15 now with Will's data, maybe there isn't a lot of change in the parasite.
Thanks, Kirk. The next question on the chat is from Ronnie Crawford, and he's asking whether it's possible to measure the linkage between genes in the host and parasite.
So just to make sure I understand the question, do you mean within the human genome to look at what's in linkage with sickle, for example, and within the parasite genome, what's in linkage with the sickle-associated loci, which I've sort of already done? Or do you mean, are you using the word linkage there to mean associations between the host and parasite? In which case, isn't that kind of what a genome-wide association study would be doing, conditioning it on the genotype of the host? Does that make sense?
Yeah, Ronnie, does that make sense to you? Yeah, Jacob has his hand up. Okay, thank you. I'll go back to the people with their hands up. The next one is Antoine. And Antoine, if you don't mind introducing yourself first, that would be really great. Thank you.
Antoine needs no introduction. No, I'm kidding.
So, hi, Antoine Classen from Montpellier. Thank you, Will. Always a pleasure listening to you. So I also like your plots showing the parasite point of view and the human point of view. And on that, it clearly shows that people with sickle, half of them are infected with PFSA plus parasites, and the other half of people are infected with parasites in which PFSA is absent. What sort of difference of phenotype do you expect in these samples? And is there any metadata available? For example, parasthenia. So I presume that the parasthenia would be higher if PFSA is present in these people.
Yeah, that's a great question. And actually, we definitely don't have any clinical metadata, but parasitemia we might be able to get. I could talk to Lucas about that, but you're right. That would be quite interesting to look at, is whether there was a difference in the parasitemia. So your hypothesis is the parasitemia might be higher in the sickle people if the parasite has PFSA. Yeah, I think that would be quite cool, actually. We might be able to get the parasitemia data. I don't think we would have any other clinical metadata available from these, but parasitemia is possible. So yeah, that would be quite cool. With a slight caveat that the sample numbers would be pretty low because, you know, as I said, only 36 people actually had sickle present in this cohort. So you're talking about like comparing 16 people if it's about 50-50. But maybe with a study that had slightly more people in it, that's definitely one analysis you could do that would be quite cool. So thanks. I'll make a note of that, actually.
Yeah, can I just add on that? Any parasitemia measurements we do have, both in the Ghana and all the other samples, there will be single point measurements. We don't have a time series. So that makes it a bit harder to really understand what's going on as well.
Thank you, Kirk. Thanks, Antoine. Anything else, Antoine? No. Okay. Thank you. Going back to the chat, thank you, Hasnan from Pakistan, to introducing yourself. Amina too has a question, and she says that your presentation is really nice. And the question is there any fitness cost for the mutations, as in the non-sickle people, the proportion are different?
Yeah, that's such a great question. And so I don't have any direct data on this. I just have my kind of nebulous thoughts. But the fact that it's totally absent in Asia, like Kirk said, and also just kind of my general intuitions about biology, I think presumably it has a fitness cost in sickle AA people to have any of these PFSA loci. And therefore, in my head, the kind of model I have is that the frequency in the parasite population will depend on the extent of the fitness cost when the parasite ends up in a AA person, the relative fitness benefit of when it ends up in an AS person, and the prevalence of AS in the human population that it's infecting. And I tried to do some modeling around that. And I actually think, I basically just didn't have time to do it, but I actually think you could model that, and that would be really interesting because you could get the data on the prevalence of sickle in every country, the prevalence of the PFSA loci in every country from PF7, and then you could use that to make some kind of predictions, at least around the ratio of what the fitness cost in AA versus the fitness benefit in AS would be, that could be consistent with the ratios of sickle and of PFSA in the parasite seen in different countries. So I think that would be quite cool, but I haven't got around to actually doing that yet.
Yeah, it's similar to sickle in humans. No malaria, no sickle. Sickle has, parasites have to be there to allow sickle to survive in the human population.
Mm-hmm.
Maybe it's the same with the PFSAs, yeah.
There are some compliments on the chat as well. Will from Dominic saying a wonderful presentation. Oliver as well. Nathaniel, great presentation. Kamran, great presentation. Bacha, excellent. Thank you. Jacob, fantastic presentation. Will, as usual.
Thank you very much.
Great presentation. And Antoine Dara is asking a question as well. So how do you explain the fact that FIK, so FIKK 4.2, association with mild malaria in Mali only? Was the definition of severe malaria harmonized across the study sites?
So two parts to that question. So the first one was, well, obviously, it wasn't only in Mali, it was also in Ghana, because Mali was the replication set. So it's in Mali and Ghana. And I still don't have any explanation for that other than what immediately leapt to Dominic Kwiatkowski's mind when I first showed him the data, which is that it's something to do with hemoglobin C. that's not really an explanation to say it's something to do with it, but it's basically this map is so striking. So what would be interesting is to get samples from Burkina Faso, where there's also lots of hemoglobin C, and to get a larger number of samples and start to try and tease out those interactions between hemoglobin S and C and the sickle-associated loci. But I mean, it's not necessarily surprising that there was no signal in Kenya because it's not present in Kenya. But what was more surprising is that there's no single signal of association in the Gambia, because there is this PFSA4 is present in the Gambia. So I meant PFSA4 is absent in Kenya, so that's why there was no signal. But yeah, why it was absent in the Gambia did puzzle me. And as you might expect, given the GWAS result, the linkage result is the same. So in Mali, in Ghana, PFSA4 is in linkage with PFSA1 and 3, whereas in the Gambia, even though PFSA4 is present, there's no linkage at all with PFSA1 and 3, which is consistent with the GWAS not finding it. So I don't really have an explanation. I don't know, biology is complicated, I don't understand it. But yeah, I think you'd need, on the population genetics side, you need to do a bigger study with more samples, better representation of countries that have different hemoglobin variants, in particular thinking about hemoglobin C in that kind of central West African area. And then there's a whole load of wet lab stuff you could do to try and unpick this apart. And I should have said, actually, PFSA4 is in 3D7. So whatever its fitness effect is, it can't be too bad because 3D7 grows okay in the lab. And also, if there's other labs out there listening who are rushing to do allelic replacement experiments of these PFSA loci, I suspect they've done them in 3D7 as a background, which already carries potentially this one. So maybe DD2 would be a better starting point because it's an Asian parasite that's not going to have any of these loci or other ones waiting to be discovered. So yeah, I don't know, basically, but it'll be interesting to find out.
Thanks, Will. Moritz, would you mind introducing yourself and then asking the question, please?
Of course I do. Hello, this is Moritz speaking. I'm calling from Portugal. I was at the crick. Super cool. I'm curious, can you explain briefly again, how often do you find the FIKK SNP alone in the absence of any of the other markers?
I don't have that specific piece of information immediately to hand. I mean, you could very roughly kind of eyeball it from the fact that the PFSA4 variant itself had an allele frequency in the general population of about 5%, I think, here in Ghana. So yeah, so 3% in non-sicklers and 33% in sicklers, but sicklers was a small percentage of the total population, so that will probably balance out as not far off 3%. So maybe 4 or 5% would be the overall allele frequency of PFSA4. And then if you combine that with, you know, the linkage of being 0.3, you know, so it's found much more often than you'd expect by chance with PFSA1 and 3, but you know, it's still quite far off being 1, so you'll still expect to see a reasonable number of parasites that only have PFSA4 and not 1 or 3, and vice versa. But I don't have the exact number to hand right away.
Okay, I'm just thinking in terms, you know, how much is it protective by itself rather than potentially allowing the other alleles to exist. If that weakens the interaction with the red cell, it could be a secondary mutation to help with that. But if it can be there alone, it argues that it may have an independent role of the other ones as well. And a second question, just briefly, if I can, have you looked in the Plasmodium rhicanovi or some of the other Laverania species, if these SNPs are found in any of the sequencing? I mean, the sequencing is not that great. We don't have as many, but it may be a bit informative.
Yeah, that's a great idea. And actually, I've not got around to doing that yet, but I should add that to my list as well.
Great, thank you.
Thank you.
Thank you very much. Next question from the chat is from John Farrow. Could you reiterate the impact of this discovery? What does it mean clinically if a parasite has PFSA4 compared to the other 3?
Let me just think about that for a second. So I think there was kind of two parts to the question. There was a general reiterate the discovery, and then there was a second part about clinical impact. And I think this would not be a study that is in the, you know, if research is kind of basic science, translational science, and clinical research, this is not in the clinical research camp. It's probably not even in the translational research camp. It's probably in the basic research camp. So I don't think this study in itself is going to be, you know, changing clinical practice. So not in any direct way. So this study wouldn't change the treatment of anyone with malaria. But what is the impact of the study is that it's starting to try and unravel the evolutionary interactions between the host and parasite within the human red blood cell. It may be that there are translational applications down the line if we better understood, you know, how Sickle works and how the parasite overcomes those mechanisms. It may present opportunities for drug discovery and so on, but that's somewhat down the line. But I think the main, you know, the main point of the study is to add to our understanding of how host and parasite interact with each other within the red blood cell. Yeah.
Thank you, Will. Inu, could you introduce yourself, please? And that's going to be our last question.
Okay, thanks, Christina. And thanks, Will, for your presentation. So I'm Inu from the Gambia, part of the genomic surveillance team here in the Gambia.
Mm-hmm.
What we see generally is that drug resistance is one of the key drivers from the parasite side, driving population structure on the parasite. So have you looked at your data in light of specific drug resistance mutations that are present in the parasite population?
Yeah, good question. So I guess the way that you might want to do that is to think about how, say, you know, different known drug resistance markers, what the frequencies are like in PFSA positive and PFSA negative parasites. That might be an interesting question. I just worry that we wouldn't really have great sample size to do that justice, and that you might then get some spurious, you know, I don't know, whatever, 76T is a lower frequency in parasites that have PFSA, but actually it's because the PFSA population is quite small, as you can see. You know, given that only like 2 to 3% of these people were sickle carriers, the overall prevalence of these are actually quite low. Most of them are less than 5% allele frequency in this population. So that's just one technical caveat. Although actually, I guess with PF7, if you use the whole of PF7, you'd have larger sample sizes rather than just using this Ghana data set. You just couldn't stratify then by sickle and non-sickle. So if you use PF7, yeah, you could look. I guess I kind of indirectly did that by looking for what samples had high linkage with the PFSA loci, and the main hits that you get are the other PFSA loci. You know, although I didn't specifically look for them, if they were in really high linkage, the drug resistance markers would have been there when I looked for high LD with the PFSA loci. But I could look specifically at what the LD was for those loci in PF7. Yeah, that would be possible, although I haven't done it. But for the reasons I just said, I don't think the signal would be super strong, or I would have already seen it.
Okay, thanks.
And actually, I guess indirectly, you might have also seen little flitters of signal in the GWAS. If the drug resistance loci were in LD with the PFSA and the PFSA associate with sickle, then you would get little bits of signal in the GWAS, which I don't think we did. But yeah.
Well, we'll need to, we are run out of time, and I'm really pleased to see so many people attending. There was 88 people will attending your journal club. So
I suspect that'll be much more than the Sunday morning slot at ASTMH. So, yeah.
Yeah, so thank you so much for your excellent presentation. There are many praises to you on the chat, and there are also a few other excellent questions. So I would encourage people who we couldn't have time to answer your questions to contact Will directly and discuss, because there are really some great ideas there. And yes, so Will, thank you so much for your time and dedication to this, and thank you so much for everybody for attending this. We will shortly be advertising the next Journal Club to happen end of October. Yes, thank you so much, everybody. See you soon.
Thanks
Bye.
very much, everyone. Bye.
Yeah.